Dit is zo de intro. Zijn om door te gaan.
Beyond the sea, somewhere waiting for me. My lover stands on golden sands and watches the ships that go sailing. Somewhere beyond the sea, she's there watching for me. If I could fly like birds on high, then straight to her arms I'd go sailing. It's far beyond the star, it's near beyond the moon. I know beyond a doubt my heart will lead me there soon. Beyond the shore, we'll kiss just as before. Happy we'll be beyond the sea, and never again I'll go sailing.
Off our coast, there are magical underwater forests. The rocky shores along the French coast show a clear zonation of different types of seaweed, including brown, red and green species. These seaweeds form an important habitat for many organisms: nematodes, copepods, ostracods, foraminifera, bryozoa, etc. Studies have shown that the diversity and abundance of associated species is partly determined by the identity of the seaweed and may be correlated with the structure of the organism. The associated organisms of 3 different seaweed species were compared and quantified. We compared meafauna and fucus, a brown seaweed, chondrus, a red seaweed, and cladophora, a green seaweed. The seaweed was rinsed, after which meafauna was counted and identified. The meafaunal composition will also be compared between seaweeds in the medialittoral and supralittoral zones. We hypothesize there will be a difference in meafaunal species composition between seaweeds of different species, and between medialittoral and supralittoral zones in the same species. In order to research this, the different species of seaweeds were collected along the French shoreline during low tide. Early in the morning, specimens were collected of the medialittoral zone, where they are submerged during high tide, and of the supralittoral zone, where they are only submerged during spring tide. Back in the lab, we then soaked 6 replicates of each sample in distilled water, incapacitating the meafauna and causing them to release the weeds. This was then filtered and repeated multiple times. The resulting meafaunal samples were rinsed with ethanol and stained with rose bengal. After letting the samples stain overnight, they were rinsed with water yet again and filtered, leaving the meafauna brightly stained. This way, they were easily observed using a binocular microscope. All samples were analyzed, and the number and species of meafauna were identified in each of them. Then we performed a statistical analysis with the collected data for each sample to determine the difference in composition between species and zones. We looked at the proportions of taxa for all samples and compared them. Finally, the Shannon index was calculated to compare species richness between samples.
Man riding the... Is that a man riding a shrimp?
We made an overview of the taxa and their abundances in the different samples and replicates. Then we looked at the proportions of taxa present in each of those replicates. When interpreting those proportions, we can see that, for example, chondrus and cladophora contain more copepods in the sea samples than in the ones collected at the shore. The sea samples of Fucus aretus have a particularly high proportion of nematodes. For cladophora, there generally seem to be more nematodes present in the shore sample. Then we made box plots of the proportions of the present taxa per sample. Chandras at sea, along with Cladophora, proportionally has the most copepods. Fucus sampled from the sea contains the most nematodes. Nauplius larvae are only found in Chandras at the shore site. Then box plots per seaweed species were made. From these, we can see that Cladophora houses more copepods at sea, but more nematodes at the shore. In the seaweed sampled from the sea, there were bivalves present, while that was never true for shore samples. For Fucus there were more copepods and ostracoids found in the shore samples and more nematodes in the sea. For Chandras, there are more copepods present at sea. The shore samples show a higher diversity in taxa like flatworms and Nauplius larvae. When comparing the two Fucus species, there are no obvious observable trends between the species. However, we do see that there are more copepods on the Cerata species, while nematodes and copepods are equally distributed. After performing tests, we found that for Chandras, the difference in abundance between shore and sea is significant for Nauplius larvae, but not for other taxa. For Fucus serratus, no taxa was significant. Cladophora showed a significant difference between shore and sea for copepods. We can conclude that there is a difference in composition between shore and sea samples for Chandras and Cladophora, which can be explained by the vastly different conditions in the medialittoral and supralittoral concerning water availability, wave stress, and salt levels. To further compare the different samples and their composition of the different species, we conducted a PCA analysis. From its output, we can conclude that there are no obvious differences in compositions between the different seaweeds. This means that the observed megafauna taxa show little preference for which species they inhabit. The Shannon Index is a measure of biodiversity. After testing for normality and for equal variances between the samples, we conducted two samples to test whether there are significant differences in that index between samples from the same species taken at the different sites. We found that only the Chandras seaweed sampled from different sites had significant differences in their diversity between shore and seaside. The sea sample was less diverse than the shore samples. This could be explained by the high disturbance rate in the medialittoral zone, since a tide coming in and the wave action may wipe away the species on the seaweeds. In contrast, there are no significant differences in the Shannon Index when comparing the different seaweed species. This means the diversity levels are equal. After measuring the evenness in all samples, we found that there are significant differences in the species Chandras for the different sampling sites. For the other species, no significant differences in this measure were found. Again, no significant differences in evenness were found when comparing the different seaweed species with each other. This means that different megafauna taxa living on seaweed species have comparable proportions in their abundances, and there are no dominating taxa. The different properties of the seaweeds thus do not seem to have an impact on which taxa are able to live on them.
Invece pasta d'acqua.
From this study, We have found that the community structure, megafauna and seaweeds differs for some species between shore and sea, particularly chondrus, while unfocus and cladophora. This is the case. This may have been a result of limited time and resources for these studies. But all is not lost. If we'll make an effort now and do more studies, there is still hope to know the true species distribution and different species of seaweeds, and planet Earth can be a better, safer place. I have been David Attenborough. Thank you all so much for watching. Please like and subscribe.